So to summarize what we did yesterday, all we basically said was we started with one 6-carbon molecule of glucose. We went through a whole bunch of steps, which we called glycolysis, metabolic pathway, and at the end of that, all we really had done was split glucose in half. And each of those are called pyruvates. So we went from one 6-carbon molecule to two 3-carbon molecules. Make sense? In this process, we also generated how many net ATP? 2. Good. So we have 2 net ATP because it costs 2 to sort of prime it and get it started, but we made 4. And we also generated 2 coenzyme, of a coenzyme called NADH, which really was NAD before. It just got reduced. It picked up hydrogens. Now we said if there was no oxygen present, this would basically go from here and shuttle off to the side and go through a process called fermentation, which would recycle the NADHs, get rid of the hydrogen, and make a byproduct like alcohol or lactic acid. But it would recycle the NAD so that you could continue to run glycolysis. Remember, glycolysis only extracted about 2% of the energy that's available in sugar. So we're now assuming we're going to go the other direction. We're going to take these and we're going to keep going. We have oxygen present. So our next step is called the oxidation of pyruvate, which has nothing to do with oxygen. It has to do with oxidation, as in oxidation is loss, losing an electron. So our first step happened in the cytoplasm. The rest of our steps are going to happen in the mitochondria. So as the pyruvates shuttle across into the mitochondria, they are actually going to lose an electron, which is why it's called oxidation of pyruvate. As a matter of fact, they're not even just going to lose an electron. I'm going to put all of this up. This is a picture of what's happening. Anybody remember what we call this functional group? There would have been an H here. Anybody recognize that?
Carboxyl.
Yes, very good. Carboxyl group. So technically what's going to happen here is an entire carboxyl group is going to break off of each pyruvate. That's actually what's going to happen. So our pyruvate is going to go from being 3 carbons, it's going to turn into something new. It's now going to become something called an acetyl group here. And an acetyl group only has 2 carbons. And then it's going to attach another coenzyme to the acetyl group that they so creatively called coenzyme A.
Oh man.
I know. It was a tough day at the office. I said, you know what, we're out of good names, we're just going to call it A. And this is actually going to be a fork in the road. I'll show you in a second what I mean by that. We also make 2 CO2s and 2 NADHs. So I'm going to give you a walkthrough of this. I know you're still writing, so I'm going to give you a sec to write, and then I'm going to show you a little walkthrough of what's happening. All right. So, so everybody's had a chance to do this. So let me show you. I think the next slide is blank. I can draw this. Oh no, this is the mitochondria. All right, so before we go to the next slide, let's talk about what's happened. So we started again, we're going to go all the way back real quick. This was glucose. It's not just carbons, I'm just only drawing the carbons. It's really C6, H12, O6. It's carbons, hydrogens, and oxygens. Our first step, glycolysis, broke this in half. What do we call these 2 3-carbon molecules?
2 pyruvates.
Right, these are called pyruvates. So we have 2 of those. Now, this is the oxidation of pyruvate step. One of the carbons breaks off and leaves. You basically exhale it. That's carbon dioxide. You exhale carbon dioxide, this is where some of that carbon dioxide is coming from. So now we've gone from a 3-carbon molecule to now 2 2-carbon molecules, and these are called acetyls. So just like this was glucose, and this is called pyruvate, this is called an acetyl. But in the same step, a little coenzyme that's called A attaches to it, and so the whole thing ends up being called acetyl coenzyme A. And now this is what's going to feed into our next step, which is the last step we're going to talk about today, which is the Krebs cycle. So our 2 acetyl groups are now going to feed into this, the Krebs cycle. By the end of the Krebs cycle, our carbons are all going to become carbon dioxide. All of our carbons and our oxygens are going to be gone. So let's go now to our next step. In fact, before we do that step, let me make another comment. The citric acid cycle. Everybody, let me tell you who's coughing in the video.
Marianne.
It's Marianne, everyone. Marianne is coughing. So the Krebs cycle happens in the matrix. You will be required to know where the matrix is. Not that computer world that we don't really live in. All right,
Well, we
so
don't know that we don't live in it.
that's true. We may live in the matrix. I'd rather live in the matrix actually than have one of those tubes in my head.
Does this happen in the matrix, like the matrix within the matrix, within the matrix, within the matrix?
I think he's the one.
This is not matrix section, okay?
What?
You didn't live in that.
No, I know what you're talking about.
Okay,
I
okay.
think JoJo's the one. I really like that drawing.
All right, isn't it beautiful? I know, and you probably shouldn't even try because yours probably will not be as pretty. All right, so this is the mitochondria, and the middle part here, this is the matrix of the mitochondria. So if I was to ask you, give you a picture on a test and ask you where the matrix was, or ask you to show me the area where the Krebs cycle happens, I would expect that you would know it's there. It's in the middle, inside. This is called the, very creative, outer membrane. I know. Guess what this one's called?
Inner.
The inner membrane.
So the picture would be like that.
Yes. This has another name. The folds are also called cristae. And then there's only one more mitochondrial part that you could be asked about, and it also has a pretty common sense name. All this white area here, this is called the intermembrane space.
Oh man.
The space between the two membranes. So that's basically a All right, so I got rid of that. So the citric acid cycle, so we started with an acetyl group, or two acetyl groups actually. It's going to turn 2 times, once for each acetyl group. So the first acetyl group is going to go in, it's going to turn, it's going to make an ATP, it's going to make 3 NADHs, 1 FADH2, and 2 CO2s. Then the second acetyl group will go in and do the same thing. So it turns 2 times because we had 2 acetyl groups from our 2 pyruvates. They're going to feed in, and so this is going to turn 1 time per acetyl-CoA, which means how many total ATP are going to be made in the citric acid cycle?
2.
How many NADHs?
6.
6. How many FADH2s?
2.
JoJo's gotcha. And how many carbon dioxides?
4.
4.
So everything doubles.
Because it turns once for each of these. Because remember we split glucose, so everything from that point on has to happen twice. Now guess what? By the end of this process, all of our carbons are gone. We started with glucose, C6H12O6. In the oxidation of pyruvate, we made 2 CO2s. Now, in the Krebs cycle, we made another 4 CO2s. If you add that up, that's all 6 carbons. All of our carbons are gone, and all of our oxygens are also gone. The oxygens are not going to add up properly, just so you know, because if you add them up, you're going to go, but every CO2 has 2 oxygens. Doesn't that make 12 oxygens? And there's only 6 here. It's because water is actually involved, and they've reduced the equation. It's a redox reaction, so if you've taken AP Chem, you know that you have to balance with hydrogens and electrons in water and all this stuff. And so just trust me, all the carbons are gone and all the oxygens are gone. All that's left are the hydrogens, and all the hydrogens are now attached to the coenzymes. So let me show you a summary of this, and then we'll be all done for today, actually. So turns 2 times, once for each acetyl-CoA. Sorry if you didn't get that. 2 times, once for each acetyl-CoA. And here's a diagram of what it looks like. You don't have to know this diagram. You're not going to be tested on memorizing all of these. But an analogy for what's going on here, and I know I tried to give you one yesterday, I think. I don't know. I gave some of my class an analogy. A Ferris wheel. But think of it this way. Think of the Ferris wheel that I'm already on the Ferris wheel. I'm sitting right here.
And then you do an origami.
Acetyl-CoA comes in, right, and it hands me the paper. I take the paper and I make my little origami and hand it off. and basically I'm still on the Ferris wheel. I get all the way back to the beginning, and I'm ready to take in another paper. Except think of it, instead of making one origami, you're taking in the acetyl-CoA, and you're giving off NADH, ATP, FADH2, carbon dioxide. So you're basically taking in the paper, fixing it, getting rid of it, and you end up back here on the Ferris wheel ready to take another paper. And that's what's happening here. So this whole complicated picture is all the individual steps. By the way, the citric acid cycle, the name comes from the fact, this one right here, citrate, citric acid. It's the first thing in the cycle. But again, you're not going to be tested on these, knowing these individual steps. Here's one where they've kind of stepped back a little bit and just shown you, here's one pyruvate. That one pyruvate becomes acetyl-CoA. Here's the CoA coming in, here's the CO2 leaving. So we went from 3-carbon pyruvate to 2-carbon acetyl, carbon dioxide left. That acetyl-CoA now goes into the citric acid cycle where it breaks and becomes 2 carbon dioxides. So now all the carbons from the pyruvate are gone. It makes 3 NADs, an ATP, and an FADH2. And then remember, there were 2 of these because glucose split in half. So now the second acetyl-CoA will come in and do the same thing. Did you have a question?
That you answered it.
Okay. And this is our last slide for today. This one really takes us all the way back to glycolysis. So if we go all the way back to glycolysis, we started with 6 carbons, right? Here's our 6-carbon sugar glucose. Pyruvates had 3 carbons each, right? So here's our 2 pyruvates. Oxidation of pyruvate, we then turned that into 2 acetyl groups, and we made 2 carbon dioxides. And then here, when these feed into the Krebs cycle, we end up with 4 more carbon dioxides. So all of our carbons are gone. But here's the important part. Here, we made 2 NADHs from NAD. We picked up hydrogens from glucose. In the oxidation of pyruvate, we made 2 more NADHs. In the Krebs cycle, how many NADHs do you remember? 6 NADHs and 2 FADH2s, which, by the way, came from FADH. It already had 1 H on it. If you add up all these hydrogens, and remember, this one, don't count 2 hydrogens, count 1 because it was already FADH, guess what? You're going to get 2, 4, 10, 12. Well, remember our original formula, C6H12O6. So all the...
10, 9,
Don't do that to me.
8.
All right, so all the hydrogens from our original glucose are now trapped in these coenzymes. Now here's my point. How many total ATP have we made up to this point?
4.
How many did we make? Yes. How many did we make in glycolysis?
2.
A net of 2, right?
Yeah.
And then in the Krebs cycle, we made 2 more. So at this point, we've only made a total of 4 ATP. Anybody remember how many total we're going to make?
36.
We're going to make 36. That means that this last step is going to make 32 ATP. And the only thing that goes into this step are all the hydrogens. So this one thing, all this prep work, all this long metabolic pathway, didn't make a lot of ATP, but it picked off these hydrogens, and now this is going to be the big payoff. All these hydrogens, when they flow into the mitochondrial inner membrane, now we're going to generate all the ATP. So the other steps had to happen. Even though they don't make a lot of ATP, they do all the prep work so that then the big payoff comes at the end. Okay? So that's where we're going to stop for today. That'll leave us with just the electron transport chain to cover tomorrow.